Biology.
So the next question is asking, define the term metamorphosis. So what is metamorphosis? So for this metamorphosis, we see that these are the stages of development whereby an insect develops from egg, larva, pupa, and adult, or some insect develop from egg, nymph, and adult. So these are the stages of development in an insect. That is what is called metamorphosis. And for these two diagrams, as you can see, we have the diagram of complete metamorphosis, whereby we have 4 stages, and then we have the diagram of incomplete metamorphosis, whereby we have three stages. So for the complete metamorphosis example, we have the housefly, mosquito, which undertakes the complete metamorphosis. And for the incomplete metamorphosis, we have organisms like cockroach, locust, grasshopper, etc. So that is metamorphosis, the developmental stages in an insect. That is what is called metamorphosis. So the next question we are being asked, differentiate between complete and incomplete metamorphosis. So for the complete metamorphosis, we see that the eggs hatch into larvae. So the eggs, they hatch to form the larvae. While in incomplete metamorphosis, we see that the eggs hatch to form the nymph. As you can look at the previous diagram, for the complete metamorphosis, we have egg, then the larvae, while in incomplete, we have egg and then the nymph. So that is the first difference we can give in this case. So the second difference, we can look at the stages, whereby for the complete metamorphosis, there are 4 developmental stages, while for the incomplete metamorphosis, there are only 3 developmental stages. The other difference we can give here, we can say that incomplete, the larvae does not look like the adult. The larvae completely in complete metamorphosis doesn't look like the adult. While in incomplete metamorphosis, the larvae looks like a small adult. Because I guess most of us, we have seen those small cockroaches. They look like the adult, but just that they are very small cockroaches. But there is now the large cockroach. So whereby if you compare this nymph, small cockroach, with the adult cockroach, they look exactly the same. Just that one is small, which is the nymph, while the adult is large. So for the other difference, we can say that in complete metamorphosis, the wings developed in pupa stage. That is what the wings developed in the pupa stage. While in incomplete metamorphosis, the wings developed through different stages. Like for instance, in the cockroach, the nymph, some of the nymph have wings already. So in incomplete metamorphosis, the wings develop through different stages. While in complete metamorphosis, so it is only and only in the pupa stage whereby the wings of these organisms will now develop. So the other difference, in complete metamorphosis, there are major changes from larvae to adult. Like you can really see that this is how the larvae looked like. A caterpillar or something. This is how the larvae looked like. But now in the adult has wings. The adult has antennae. The adult looks like this in comparison to the larvae, which looked like that. While now in complete metamorphosis, there are minor changes from the nymph to adult. Just that the nymph is a tiny adult, while the adult is large. So they look exactly the same. So there are minor changes between the nymph and the adult. So the other difference you can say, in complete metamorphosis, or maybe the organism. So you can say that in complete metamorphosis, we have organisms such as the housefly, we have organisms such as the bees, while in incomplete metamorphosis, we can give examples such as the cockroach, the grasshoppers, the locust, the cricket, etc., and many more. So the next question, follow-up question, letter C, is asking, say the importance of metamorphosis. So what is the importance of metamorphosis? Why should why should insects go through different developmental stages? Why should they go through different stages of development? Why is it that they should not grow all of them in one place? Why should they grow in different stages? So that's what this question is asking. State the importance of metamorphosis. So the first one we can say, to reduce competition. So this Process of metamorphosis reduces competition, whereby you see that organisms have different life stages which take in different nutrients. So the eggs, they use the nutrient inside the shell. The larvae, they use the different nutrients in order to develop. The pupa, they use different nutrients to develop in the pupa stage. And adult also eats another different nutrient from the egg, larva, and the pupa. So this reduces competition, especially for food and space. So the other importance, it's for growth and development, whereby during these different stages, organisms are able to develop. The different organs in these organisms are able to reorganize themselves in order now to form a complete adult, which is mature and can be able to reproduce in order to form the different organisms of the same species. Apart from that, the other importance of metamorphosis is to avoid predation. Because you see, different stages have different predators. There are predators which feed on the eggs. There are different predators which feed on the larvae, pupa. There are different predators which feed on the adult. So the other importance of metamorphosis is that the organisms are able to avoid predators, whereby different stages have different predators. As this increases their great chances of survival in the environment. So, question next, follow-up question letter D is asking, give 3 examples of insects which undergo complete metamorphosis. You have given them. And if you look at this diagram, so this diagram gives us examples of different organisms which undergo metamorphosis. We can see that we have the wasp, we have the butterfly, we have the bees, we have the moth, we have the flea which feeds on maybe the dogs, etc. and many more. Also the ant and many more. So these are organisms which undergo complete metamorphosis, while these are not the organisms which undergo incomplete metamorphosis. We have the termite, we have the mayfly, I think, dragonfly, etc., etc. Yeah, so these are organisms which undergo incomplete metamorphosis, while the other ones, they undergo complete metamorphosis. So to be on the safe side in exam, maybe just get 2 or 3 and you're good to go. Maybe just understand 2 or understand 3 and then you are good to go. So the next question is asking, say the advantage of complete metamorphosis over incomplete metamorphosis. So what is the advantage of complete metamorphosis over incomplete metamorphosis? So the first advantage, you can see that there is no competition for food. So completely the essence of competing for food has been cancelled out, because we see that in incomplete metamorphosis, the nymph feeds on the same food as the adult is feeding on. Because remember, in incomplete metamorphosis, the nymph looks like a small adult. So the food that the nymph is eating is exactly the same food that the adult is eating. So in the complete metamorphosis, we see that we have egg, whereby the embryo feeds on the nutrients in the egg. Larvae feeds on different things. Pupa feeds on different things. Adult feeds on different things. So there is no competition in complete metamorphosis. While in incomplete metamorphosis, there is competition, because the adult and the nymph, they feed on exactly the same thing. So that is the main advantage of complete metamorphosis over incomplete metamorphosis. So also we see that in these 2 types of metamorphosis, the predators for the adult, that is for the incomplete metamorphosis, the predator for the adult can also feed on the nymph, because the nymph resembles a small adult. So the predators can feed on the nymph, while in complete metamorphosis, there are different predators for every stage. So that is the advantage of complete metamorphosis over incomplete metamorphosis. In complete metamorphosis, there is no competition for food, and also we have different predators for every stage. While in incomplete metamorphosis, there is competition for food between the nymph and the adult. Also, they share the same predator, that is the nymph and the adult. So question next, we are being told, use the graph below to answer the questions that follows. So as you can look at that graph, we have the height and then we have the time in days. That is the graph. Of course, if you look at this graph, this is the graph of intermittent growth. It's a growth curve, whereby, remember, we said we have 2 growth curves in biology that we studied. We say that first of all we have the sigmoid curve, and then now we have the intermittent. Now, question number 1, we are being asked, name the graph. So this graph is called the intermittent growth curve. So we see that the horizontal lines are called the insta and indicate point where no growth is taking place. That is the horizontal line. No growth is taking place in these regions. That is called the insta. So whereby we see that the vertical lines show rapid growth, whereby the exoskeleton has been shed off, thereby the organism is going to grow very much rapidly during that region. So in these horizontal lines, which are called the insta, molting should take place in order to allow now for growth to take place in now the vertical regions that we have. So for this intermittent growth curve, mainly it takes place in organisms which have the exoskeleton, whereby this exoskeleton must be shed off, and then after shedding now off this exoskeleton, now the organism is able to grow. So after now this organism has grown again, the exoskeleton grows, growth stops, that is another insta, and then when the organism wants to increase in size, the exoskeleton sheds off, and then this organism is again able to grow. So don't forget that we have the two growth curves. We say that we have the intermittent growth curve, as you can see. This is mainly for the arthropods, and then now we have the normal sigmoid curve for other organisms. For in this normal sigmoid curve, remember we have the different stages which explain the stages of rapid growth to the stages of decline in growth. For the intermittent growth, remember that we say that we have these regions which are called the insta, whereby no growth is taking place. So part B of the question is asking, what is the name given to part labeled X? As you can look at the diagram, we have that part. Yeah, we have that part which is labeled X. So what is the name given to this part or region labeled X? So the name given to that stage here, molting mainly takes place. So we can call it the molting stage, because in here the organisms, the insect is molting. Molting represents shedding of the exoskeleton. So the organism is molting in order to allow for growth to take place. Question C is asking, state the importance of the part labeled X. So what is the importance of the part of this part which is labeled X? So this part labeled X, we see that rapid cell division leading to growth of the organism is taking place. There is a very high amount of cell division taking place in this region. Also in this part labeled X, we see that there's the renewal of the damaged exoskeleton. So the regions of the exoskeleton that were damaged are being renewed, are being formed again. Apart from that, you can say that there is also the removal of the different parasites and pests that were embedded in the exoskeleton. Because in this exoskeleton, also there are parasites which live in the exoskeleton. There are also pests which feed on this organism that live in the exoskeleton. So during the process of molting, which is the shedding of the exoskeleton, these different parasites and pests are removed from this exoskeleton. So question next, we are being asked, describe the effect of the following hormones in insects. So the different hormones which are found in insect which lead to this process of molting, etc. So describe the effect of the following hormones in insects. So the first hormone is molting stimulating hormone, MSH. So what's the function or what's the effect of the molting stimulating hormone? So this molting stimulating hormone stimulates the production of molting hormone. So this is the one which now facilitates the molting hormone to be produced. But what is the function of this molting hormone? So this molting hormone stimulates the degeneration of the exoskeleton. So it mainly stimulates the degeneration and shedding. I think that's the best word to use. The degeneration and the shedding of the exoskeleton, which now will lead to the process of metamorphosis. So the exoskeleton will now start breaking down slowly due to this molting hormone, which will finally lead to the process of metamorphosis. So the next one, we are being asked juvenile hormone. So what is the function of juvenile hormone? So the juvenile hormone, we see that it maintains the larval stage in incomplete metamorphosis. So it is the one which is used to maintain and hold on to this organism, so that the organism, can be able to hold on during the larval stage and then make it to further and further continue development to form the pupa. So that's the first one. So So the second function of the juvenile hormone, you can say that it prevents premature metamorphosis from taking place, so that this organism will wait for the right time and then now begin the process of metamorphosis. The exoskeleton to now begin shedding so that metamorphosis process can take place. Then finally, the juvenile hormone, you can say it inhibits and stops the process of metamorphosis. This is the one which now puts to an end the process of metamorphosis for this organism, to stop the process of metamorphosis so that now it can continue the other stages of development. Yeah, so the next question it's asking, use the diagrams below to answer the question that follow. And I can see these cotyledons. I know we have here the types of germination. So the first question is asking, identify the types of germination A and B. So germination type A, this is the epigyl germination. Why is it the epigyl germination? Because you can see the cotyledons are coming out of the soil during germination. Remember in the previous question you said, if cotyledons emerge out of the soil as the seed is germinating, that is epigyl germination. And this is exactly the same thing. So it is epigyl germination because the cotyledons are coming out of the soil as the seed is germinating. So part labeled B, we see that diagram B, this is hypogyl germination. Why is B hypogyl germination? Is because the cotyledons are inside the soil as the plant is germinating. Since they are inside the soil as the seed is germinating, this now forms the hypogyl germination. Remember again, let me remind us, for these two types of germination, epigyl germination, hypogyl germination, how do you differentiate between them? Check the position of the cotyledons. So the cotyledons are the ones which are going to tell you that this is epigyl germination, this is hypogyl germination. Just check the position of the cotyledon. If they come out of the soil in germination, epigyl germination. If they remain inside the soil during germination, hypogyl germination, and don't forget. So the next question is asking, the diagram below is a part of the stem. As you can see, the diagram below is a part of the stem. Name the part labeled A and E. So this part of the stem comes from what we studied in Form 2, the topic of transport in plants and animals, the subtopic of the stem. So this diagram emerges exactly from that diagram because it is just a section which has been cut, as you can see. It is a section which has been cut from the main section that we studied in the monocotyledonous stem, dicotyledonous stem. So question part A, name part labeled A to E. So what's the name given to part A? The outermost part of this stem. So of course this is the epidermis. That is the name given to the outermost part of this stem. That is the epidermis. So the next one, we are being asked to name the part labeled B, which now is going to surround the phloem. So this part labeled B, which surrounds the phloem to the outside part, it is always the sclerenchyma. So this part B, again, remember, this is the sclerenchyma part, and then automatically part C now becomes the phloem, because it's the phloem which is found on phloem, the vascular bundle which is found on the outer side, and then on the inner side, remember, we have the xylem. So part labeled C, that is the phloem. Then the next one is part labeled D. Part labeled D, that is the cambium which surrounds the vascular bundle. And since we have the cambium in this diagram, then this diagram is of a dicotyledonous stem, because it's the dicotyledonous stem which has the cambium which passes through the vascular bundle. How did I know it was a cambium? Because it is passing through the vascular bundle. That is how you know that is the cambium. Then finally part labeled E, that is the pith, which is used for storage of water, nutrients inside the stem. So part B of the question is asking, state the function of the part labeled B and D. So state the function of the part labeled B. B is the sclerenchyma. And when we are discussing cell specialization, we say that sclerenchyma is a modified plant cell which is used for support. So it is used to provide support and strength for the stem or for the system where it is found. And that is the function of the part labeled B, is to provide support and strength for the stem system. What about the part labeled C, which is the phloem? So this part labeled C, it is used to translocate food materials within the plant system, because part labeled C, that is the phloem, it is used to translocate food materials within the plant system. But what about the part labeled D? That is the cambium. So the part labeled D, on this other hand, we see that cambium is used for secondary growth. Secondary growth means to increase the width or the girth of the plant. Remember, we had just discussed that. So to increase the width and the girth of the plant. That is the function of this part labeled D. So question next is asking, say the meaning of the following terms. So the first term we are being asked, metamorphosis. So what is metamorphosis? What's the function, or what is the definition of metamorphosis? So this metamorphosis, we see that this is where an organism develops through stages from egg to adult. That is metamorphosis. So there are different stages which take place for this organism to develop from egg to adult. Whereby, remember, in complete metamorphosis we have egg, larva, pupa, and adult. While in incomplete metamorphosis, remember, we have egg, nymph, and adult. That is the definition of metamorphosis. The development of insects from egg to an adult, or the development of organisms or insect through different stages. That is what is called metamorphosis. So apart from that, question next, we are being asked to define ecdysis. So what is ecdysis? Ecdysis is another term to define molting. That is ecdysis. So simply for this ecdysis, you can say that this is molting, whereby an organism sheds off its exoskeleton to form a new one in the process which is called metamorphosis. So this simply is metamorphosis. Ecdysis, metamorphosis is exactly the same thing. So if you hear ecdysis, if you hear molting, they represent exactly the same thing. So question next, we are being asked, below is a diagram of a dicot root section. Name the zones A to D. So the name given to the region marked A, we have the region of permanent tissue. That is the name given to region labeled A, the region of permanent tissue. What about the region labeled B? So the region labeled B, this region is called the region of cell elongation. And also the region labeled C is also the same, the region of cell elongation. And then finally the region labeled D, this is the region of cell division, whereby we have now the apical meristem. So the region of cell division, whereby we have too much auxins, or we have auxins which facilitate cell division, like in order to increase the growth of this root down the soil. Those are those regions. So next question is asking, what is the role of vascular cambium in the growth of flowering plants? So what is the function of the vascular cambium? Vascular from the vascular bundle. Cambium, which is found embedded in the vascular bundle. So it's the function of the vascular cambium in the growth of flowering plants. So you see that this region contains cells which divide by the process of mitosis, and then they produce secondary vascular bundles. Yeah. So in this region we have very many cells, and then these cells are able to divide by mitosis. Remember, mitosis is the division of body cells or somatic cells, and then as these cells are dividing, the cell is going to increase, the stem is going to increase in girth, leading to secondary formation of secondary vascular bundles. That is it. So due to this division, we are going to get now secondary vascular bundles. So this secondary vascular bundle, remember, their function is to provide support, is to offer transport of nutrients and transport of water minerals also within the plant, and also their main function is to form the bark and the woody part of the stem. So those are the functions of the vascular bundles that the secondary vascular bundles lead to. So apart from that, the next question is asking, why is oxygen necessary in seed germination? Anytime you'll hear oxygen being mentioned in line of germination, energy. The main thing here that's going to flow is energy. So for the answer to this, you can say that oxygen is used in the process of aerobic respiration during the process of germination in order to produce energy for the seed to develop. That is it. Anytime you hear oxygen in photosynthesis, respiration, nutrition, which process? Oxygen is used in respiration. So in this case, oxygen provides energy so that now this seed can continue the process of germination. So don't you forget, again we said we have two types of respiration: aerobic respiration, whereby oxygen is used for the plant to make its own food. After that we have anaerobic respiration, whereby no oxygen is used for the plant to make its own food. So we discussed them in the previous class, you can check them out. So question next, we are being asked, differentiate between primary growth and secondary growth. So we have differentiated this in the previous questions, but again we are revising, it's no harm to go through it again. So you see that primary growth occurs at the apical meristematic regions, while secondary growth occurs at the cambium regions of the plant. So again, remember we said primary growth increases the height of the plant, the shoot to go up and the root to go down, while secondary growth increases the plant width. So don't forget that, the difference between primary growth and secondary growth. Primary growth is mainly brought by increase in cells at the meristematic regions, while secondary growth at the cambium regions. So it is what it is. So question next is asking, define intermittent growth. So what is intermittent growth? So we have just looked at the intermittent curve, but now we are being asked, what is intermittent growth? So this term intermittent is an English word meaning not continuous, something which happens and then stops suddenly. That is the meaning of intermittent according to the dictionary. It occurs in irregular intervals. That is intermittent. Maybe for instance, we can talk about the monthly periods that some females have. If monthly period is irregular, we can say that the girl is having intermittent monthly period. So again, remember, if we say they have intermittent monthly period, it will mean that the periods are not regular. They happen, then they stop suddenly, then they happen. That is intermittent, irregular intervals. So now, since you are talking about intermittent growth curve, the intermittent growth curve therefore means that it refers to growth which alternates, or growth which does not occur in regular interval. So growth which alternates between, now there is growth, then there is no growth again. There is growth, there is no growth again. And that's why this curve is showing the vertical lines. Remember, there is growth, horizontal line, there is no growth. Vertical line, there is growth, horizontal line, there is no growth. That is what is referred to as intermittent growth curve. So growth alternates between growth periods and no growth period. So the next question is asking, give one phylum in kingdom Animalia where all members exhibit intermittent growth curve. So one organism. So give one phylum here. Give one phylum in kingdom Animalia where all members exhibit intermittent growth. Of course, that is phylum Arthropoda, comprising of where insects are found. So it is phylum Arthropoda, since organisms in phylum Arthropoda possess an exoskeleton preventing continuous growth. So whereby this exoskeleton must be shed off so that growth will now take place. So the answer is phylum Arthropoda. Why? Because organisms in this phylum, they possess an exoskeleton which must be shed off in the process of molting or ecdysis in order to facilitate the process of metamorphosis. So the next question is asking, name the plant hormone whose effects are described below. So name the plant hormone whose effects are described below. So letter A is asking, in high concentration, they close the stomata. In high, remember the question is high concentration. So in high concentration, they close the stomata. Of course, this is abscisic acid. It is the only hormone which facilitates or which plays that role, or which facilitates the process of closing the stomata. Mainly it takes place during the night. During the night, the stomata are going to close, while during the day, the stomata are going to open. So it is the function of abscisic acid to facilitate this process. But what are the functions of abscisic acid? Maybe if we can try to remind ourselves. So you see that it inhibits growth in plants. Also remember we say that it induces closing of stomata, what we have just said. And then maybe apart from that, you can add more and say it inhibits the development of the lateral buds. So you have just gone through the functions of abscisic acid in the previous question, but those are just a few. So part B of the question, we are being asked, induces parthenocapy. So which hormone induces parthenocapy? In the previous question we say this, gibberellins, and again here we are still going to say this, gibberellin hormone. So just a few functions of gibberellin hormone. We say that gibberellin, it induces flowering, or it facilitates. If induces seems a tough English, you can say facilitates flowering in plants. So it facilitates flowering in plants. Apart from that, it also promotes cell division in the plants. Apart from cell division, remember I say that it is also used to break seed dormancy. So if a seed is dormant, if you spray it with gibberellin hormone, it is going to break the seed dormancy. Apart from that, you can also say it initiates parthenocapy, as the question is asking. So part C of the question is asking, inhibits development of side branches from, of course, this is the auxin or indole acetic acid. Where auxins are, the plant is going to grow up and down. The roots are going to grow down, the shoots are going to grow going up. That is what we are saying, that this hormone inhibits development of side branches. It prevents the development of side branches, but promotes the development of the shoot to go up and the roots to go down. That is auxin hormone, whereby in this term we called it, what did we say? Apical dominance. We say it's called apical dominance. So the apical meristems are going to dominate growth. Apical meristems of the root will dominate the root to grow down, of the shoot will grow going up. We call it apical dominance. So just a few functions of the auxin. We say that auxin promotes growth of adventitious roots that we saw the diagram. Also we say that it promotes flowering in plants, it promotes apical dormancy, it breaks seed and bud dormancy, it initiates cell division of the meristematic regions of the cambium, among others, very many more functions of auxins. Yeah, the next one is that diagram. So the diagram, the question is asking, define the following terms. So the first one is insta. So you have gone through this diagram, another one, but this another diagram is still okay. So the first question, define the following terms. Insta. So what is insta? So in the previous question, remember we said that insta is the stage of development of an insect between the different molds. So the stage of development of an insect between the different molds, that is what we called an insta. How do you identify an insta? So the vertical line in the intermittent growth curve, that is always the insta. That vertical line is always the insta, and never forget. So define the following terms. The next one we are being asked, apical dormancy. So for the apical dominance, you can say this is the faster and the taller growth of stem than the side branches. So the stem is going to grow taller, faster than the side branches. That is apical dominance. Apical dominance from apical meristems from auxins, which facilitate the vertical growth of the plant. So that is what you are calling apical dominance. So the plant is going to increase in height faster than increasing in the gap. So part C of the question is asking, explain what happens in pupa phase of metamorphosis. So what happens during the pupa phase in metamorphosis? So during the pupa phase, we see that the larval tissues break down as the materials are used to synthesize adult tissues. So the larval tissues are going to break down. If you can look at this diagram, so all the larval tissues are breaking down, in the organism as different materials used to synthesize adult tissues are being formed gradually in development of the pupa phase. So that is it. So the pupa is going to develop, more of the cells and tissues of the pupa are going to form in order to propel now this pupa, now slowly and gradually, now to begin forming adult properties as the organism is growing. Question next is asking, state the two types of metamorphosis. So what are the two types of metamorphosis we have seen? We have complete metamorphosis, we have incomplete metamorphosis. In complete metamorphosis, we have 4 developmental stages. We have egg, larva, pupa, and adult. In incomplete metamorphosis, we have egg, the nymph, and finally the adult. So don't you forget that. So next question is asking, name the part of the seed whose growth brings about epigyle germination. We have gone through epigyle germination a million times and hypogyle, but now this question is asking, name the part of the seed whose growth brings about epigyle germination. So for the epigyle germination, remember we said we have the hypocotyl. For the epigyle germination is hypocotyl. Remember, letter E, letter H. Letter H, letter E. For the epigyle germination, hypocotyl. For the hypogyle germination, we have the epicotyl. And for the hypocotyl, remember we said that this is germination, the epigyle germination, we say that this is germination which takes place above the ground, while hypogyle germination is germination which takes place below the ground. As you can look at these 2 diagrams, they try to emphasize epigyle germination and hypogyle germination. So remember, epigyle germination we have hypocotyl. Hypogyle germination, remember we have the epicotyl. So the question next we are being asked, state the role of oxygen in germination. Of course, oxygen, we have gone through it a million times. Oxygen is used in respiration. Oxygen is used in oxidation of food in order to produce energy. So those are the functions of oxygen: respiration and also oxidize the food in order to provide energy and nutrients for the plant. So the next part of the question is asking, name the hormone in plant responsible for part A, seed dormancy. So which hormone is responsible for seed dormancy in plants? Abscisic acid. So in that question, if you have been asked that question in exam, the answer you give is the abscisic acid. So part B of the question is asking stem elongation. Which hormone is used in stem elongation? Gibberellin hormone is the one which is used in stem elongation. So auxin, yes, but the best answer to give in this place is the gibberellin hormone. Why didn't we say auxin as the first choice? Because auxins are mainly found on the meristematic region. In the stem region, the main hormone which facilitates stem elongation is the gibberellin hormone. And that serves as our answer in this step. So question next is asking, state the role of enzymes in seed germination. So state 3 roles of enzymes in seed germination. So that the roles of enzymes in seed germination. So the first one, of course, just like the human being, just like the mammals, is still the same as for the plants. So hormones, they facilitate synthesis of new organelles in the cells of this seed which is germinating. Also, hormones, the enzymes facilitate the process of respiration. They also facilitate the process of active transport within the plant, and also they are used to hydrolyze the food from the food reserves, which is the cotyledon and the endosperm. So at least those are the functions, the few functions of hormone that you can give in this step. So the next question is asking, study the graph below. Of course, another graph of intermittent growth. Study the graph below and answer the question that follows. So part A of the question is asking, what is the name given to this growth curve? Intermittent growth curve. So the other name of intermittent growth curve, by the way, you can also call it discontinuous growth curve. So you can call it intermittent growth curve or discontinuous growth curve. It's still the same thing. Why discontinuous? Because growth is occurring, then stopping, then occurring again, then stopping. So you can call it intermittent or discontinuous. If you give an exam, utakuwa tu sawa. So part B of the question is asking, name the phylum of organisms which undertake such growth. We said in the previous question, we have phylum Arthropoda, which facilitates the process of intermittent growth. Why intermittent growth in phylum Arthropoda? Because they contain an exoskeleton which must be shed off so that the organism grows, and then the exoskeleton forms again, growth stops. The exoskeleton is shed off, growth again takes place. So it is in phylum Arthropoda because they possess an exoskeleton. Part C of the question, name process labeled X. So that process which is labeled X, that is process is called the instar. not the instar, that process is called molting, sorry. That is the process of molting, which facilitates the shedding of the exoskeleton in order to permit growth to take place. So question next is asking, state two structural factors that cause seed dormancy. So structural factors means within the seed, not environment, within the seed itself. So state three structural factors which cause seed dormancy. So the first one, you can say tough seed coat. So if the seed coat is tough, no water is able to get in and hydrolyze the food to make the embryo obtain the food and start the process of growth. So that's the first one, tough seed coat. We can also have waxy seed coat, which will prevent water and nutrients from entering into the seed micropyle to facilitate the process of growth. Apart from that, you can also say impermeable seed coat. Yes, the seed coat looks moist, but nothing can be able to enter. So you can talk about tough seed coat, waxy seed coat, impermeable seed coat. They can also serve to function in this process. But for the environment, we can talk about low oxygen in the environment may cause seed dormancy, low temperature in the environment may cause seed dormancy, low moisture content in the environment, among others. And also germinating inhibitors in the environment can also cause seed dormancy. These are for the environment, but for the structure within the seed itself, we can talk about the seed coat, tough seed coat, impermeable seed coat, waxy seed coat, etc., which will lead to seed dormancy. So the next question is asking, name the tissue responsible for secondary growth in flowering plants. So which tissues are responsible for secondary growth in flowering plants? So we have already discussed this. We'll just go through them. We have the vascular cambium, which produces the secondary phloem and the secondary xylem. Apart from that, you can also say that we have the core cambium, which produces cork cells forming the back of the stem. And then finally, we say that we have the intercalary cambium, which contributes to the elongation of the stems and leaves. Apart from that, the main function is it contributes to the elongation of the stem regions within the plant. As you can look at this diagram, so this is the diagram trying to explain the different regions that you have just discussed. The cambium, the vascular cambium, we have the core cambium, and now the internal structure as how these cambiums have been arranged within the stem. Because you can see this tree has been cut like that, then that is the part whereby the tree has been cut. So the diagram at least shows where these cambiums can be able to be obtained or can be able to be found within the stem region for better understanding. So question next is asking, suggest why dry mass is more reliable measure of plant growth than fresh mass. So suggest why dry mass is a better and more reliable measure of plant growth as compared to fresh mass. Dry mass means the body of the organism minus water. That is it. Just the body, the fleshy part of the organism, utoe maji yote. That is the dry mass. The fresh mass ni, we have the body of the organism, but iko moist, kuna maji. Like we human beings that we are living, plants, any organism living, iko na maji ndani. That is the fresh mass. But if you take this organism, maybe you roast the organism so that all the water will evaporate. That is which is called the dry mass. So this question is asking, suggest why dry mass is more reliable measure of plant growth as compared to fresh mass. So for the dry mass, you see that dry mass includes only organic matter, which would give accurate mass of the organism, while for fresh mass, it is the mass of the organism together with the mass of water. You see, so for the dry mass, it's only the mass of organic matter of the organism. Only the organic matter. But for the fresh mass, it is the mass of organic matter of the organism together with water. So for the dry mass, it is better measure of plant growth because we are able to measure how much of the organism how much cells and tissues of the organism have increased as compared to fresh mass, whereby we are going to measure the mass of the organism together with water, which will give us a wrong reading. So question next is asking, say the role of vascular cambium in growth of flowering plants. Vascular cambium, we have said, the vascular cambium divides by mitosis in order to form secondary xylem and secondary phloem. Secondary xylem mainly provides support for the plant, while secondary phloem, it forms the bark and the storage tissues of the plant. So that's the function of secondary xylem and secondary phloem, just to expound more on what we have studied in the previous. So question next, we are being asked, why is oxygen necessary for germination? We have said the function of oxygen in germination is that oxygen is used for the process of aerobic respiration to provide energy and also to oxidize the food substrate in order to produce energy. So we say that germination is the process whereby seeds develop to form a new plant. That is germination, whereby a seed will develop to form a seedling and then finally to form now the plant. So remember the requirements for germination that we said. For germination to take place, there must be water, air, not really air, it's best to say oxygen, but if you can also say air. So we have water, oxygen, optimum temperature, and viability. The seed must be viable. The embryo must be mature. The seed must be alive. The seed must not be sick in order for optimum germination to take place. So the next question is asking, differentiate between primary and secondary growth in plants. So for the primary growth, remember, it mainly deals with the apical meristems to increase the plant height, while the secondary growth, it takes place in the stem in order to increase the plant girth. Again, that diagram is just to remind us of the cambium regions, the cambium regions which mainly facilitate the process of increase in the girth. The increase in the girth, which is mainly brought about by the cambium cells of the plant. So the different cambium cells which increase now the width of the plant. So for the primary growth, it increases the height of the plant. For the secondary growth, it increases the width of the plant. So the other question, so remember we are using different books to get different questions. So there is another question here we are being asked, differentiate the term intermittent growth. So for the intermittent growth, remember we said intermittent growth, this is the growth period which is followed by no growth occurring in insect. This is another diagram to show the intermittent growth. Growth occurs, growth stops. Growth occurs, growth stops, like that. That is a diagram to show the intermittent growth. So question next, we are being asked, give one phylum in kingdom Animalia where all members exhibit intermittent growth. We say that is phylum Arthropoda. What is the reason? It's because they contain an exoskeleton. So in this exoskeleton, it must be shed off in a process called molting or ecdysis in order for growth to take place. So remember we say that molting is the process whereby an insect shed off its exoskeleton in order to allow the process of growth to take place. That is what we call molting. And the next question is now asking, name the plant hormone whose effects are described below. So the first effect, closing of stomata in high concentration, we say this was abscisic acid, induces parthenocapy, we say this gibberellin, and then inhibits development of side branches, we say that was auxin. So let's go to the next question. It's asking, below is a diagram showing half of a seed. As you can see the yellow diagram, so below is a diagram showing half of the seed. So what is the role of the part labeled E in germination? So the part labeled E in germination, we say that this is the cotyledon which acts as a source of nutrients. When this part is hydrolyzed with water, so if it is hydrolyzed, so it is going to release the nutrients, then the plumule is going to, the embryo is going to use these nutrients in order to facilitate growth and development. So as the embryo grows, it's going to form the radicle which will eventually form the roots. It's going to form the plumule which will eventually form the shoot. So the next part of the question we are being asked, name part C and D. So part C, of course, that's the plumule. Then part D, of course, that is the radicle. So the plumule, remember, it forms the first leaves which facilitate the process of photosynthesis. The radicle will now form the roots which will anchor the plant in the soil and also which will now facilitate the process of absorbing water and mineral salts. So the question next we are being asked, use the diagram below to answer the questions that follow. As you can see, we are talking again about the types of germination: hypogel germination, epigel germination. So the question is asking, name the type of germination in the diagram. So remember, the cotyledons are underground. If the cotyledons are underground, that is hypogel germination. So if the cotyledons are underground as the plant is germinating out of the soil, that is called the hypogel germination. So the part B of the question is asking, name part G. So what's the name given to the part labeled G? That is the coleoptile. So that is the name given to the part labeled G, that is coleoptile. So part C is asking, state the function of the part labeled G, that is the coleoptile. State the function of the part labeled G. So this part labeled G, we see that is the coleoptile, which protects the plumule as it emerges out of the soil. That is the function of the coleoptile. It protects the plumule as the plumule is emerging out of the soil. So that is it. So the follow-up question we are being asked that the diagram below represents a maize seedling. Name part F and G. So the part F, we have said that the part F is coleoptile. It's exactly the same as the previous diagram. So that part F, that is the coleoptile, which protects the plumule as it emerges out of the rough soil. That is coleoptile. What is the name given to the part labeled G? That is a seed. That is a developing seed. You only say that because, of course, it's a seed. Nothing else. Yeah, that's the name given to the part labeled G. You can say germinating seed. Yeah, it's best to say germinating seed. Avoid saying a seed because it's a seed which is germinating. It is producing the early roots and the early shoot. So that is a germinating seed. Part B of the question you are being asked. State the function of the part labeled F, G, and H. So state the function of the part labeled F, G, and H. So the part labeled H, we see that that's the coleoptile, which protects the plumule as it emerges out of the soil. So what is part G? In part G, remember, we have the cotyledon, we have the endosperm. So we can answer in line of that. You can say it acts as a sort of source. It acts as a source of nutrients for the developing and germinating seed. So that is the function of that part. So it provides nutrients. It provides nutrients for the germinating seed. And then part labeled H, those are the early leaves. So the main function of those is, of course, photosynthesis, because those leaves are green. So their main function is photosynthesis. So part question labels, not question labels, so question letter C. Name the type of germination. This germination is hypogel germination. Why is it hypogel germination? Because as the seed is germinating, the cotyledons are found underground. That is the reason it is hypogel germination, because the cotyledons are under the ground as the seed is germinating. So question next, you are being told 3 specimens of dicot seed were soaked in water and left at under different conditions. So 3 specimens of dicot seeds were soaked in water and left under different conditions. So we can see the question is saying we have specimen A, specimen B, C. So we have specimen A at 25 degrees Celsius was left in the dark. Specimen B at 25 degrees Celsius was put in the light. And specimen C at 0 degrees Celsius was put in the light. So as you can look at this diagram, this is the picture whereby we have plant A, B, and then finally we have plant C as per the diagram. So the question is asking, explain the observations made for each specimen after 4 days. So what observations were made for each specimen after 4 days? So for this diagram ABC, we can see that in specimen letter A, there's the formation of, we can call it an in biology they call it etiolation and weak stem. So there was, the plant organism was etiolated since it also produced weak stems as the plant grew in the absence of light. So for this etiolation and weak stem, it was as a result of the plant growing in absence of light. Thereby, it produced a very weak stem. So the stems were very weak due to the absence of light. But in specimen B, we see that it was placed where there was much light, there was enough light. So in this specimen B, we see that there was optimum light, so the plant grew strong and the stem of this plant was strong. Yeah, because also light comes with temperature. That optimum light UV, which is used by the plants in order to facilitate metabolic processes, comes with optimum temperature. So for this specimen B, we see that optimum light and temperature provided, and therefore this plant stem grew very strong. While in letter C, so in letter C we see that it was at 0 degrees Celsius in the light, that is at freezing point. So at low freezing point, the temperatures made the plant's enzyme to be inactive, which led to very slow growth. So since there was very low temperature, the enzymes in this plant were not active. Since the enzymes in this plant were not active, so there was very slow growth, which was exhibited by this plant. So that is letter A. It grew very tall and very weak. Letter B, it grew very strong because of optimum light and temperature. Letter C, freezing point, enzymes inactive, so the plant was stunted. So the next question is asking, list two observable differences between specimen A and specimen B. So what are the differences between specimen A and specimen B? Now observable, what we can see. So you can see that leaf of B has larger leaves as compared to leaf labeled A, because you can just see leaves of plant B are larger than the leaves of plant A. That is one difference. Remember, the question is asking what you can see. So the other part of the answer, we can say that the stem of B is thicker than the stem of A. Apart from that, you can say that plant B is shorter than plant A, because this is exactly just what you can see. And then apart from that, you can say, let's look at the diagram. Plant B is stronger than plant A, because you can see plant B is growing vertically up, while plant A is kind of wavy. We can say that plant B is stronger, it looks stronger than plant labeled letter A. So the next question is asking, state three functions of hormones below. The first one is auxin hormone. So what's the function of auxin hormone? So we can say auxin, it promotes apical dormancy. We have explained that already. Apart from that, it promotes the growth of adventitious roots. So remember, we say these are adventitious roots. What are adventitious roots? Adventitious roots are roots which emerge from anywhere in the plant away from the root tissue. So if roots emerge from the stem, those are called adventitious roots. So remember that roots emerging from anywhere apart from the normal root tissues, those are the ones which are called the adventitious roots. So apart from that, you can also see that the other functions of the auxin, you can say it leads to the formation of woody tissue. They induce parthenocapy, whereby parthenocapy, this is the development of a seedless. This is the development of fruits without fertilization. Yeah, development of fruits without fertilization. We call it parthenocapy. Also, it promotes flowering in plants. So the next question is asking, study the diagram below and answer the questions that follow. As you can look at this diagram, we have diagram labeled M, then we have letter N on the top and bottom, then finally we have letter P and letter Q. This may talk about cell differentiation modification, because letter M, so you can see that letter M finally is forming letter P, finally is forming letter Q. So let's look at the question. Part A of the question, Roman 1, cell M becomes cell N. So name the process whereby cell letter M become cell letter N for no. So this is through cell division that you can see that cell letter M has multiplied to form two cells of the same kind. That is cell division. In this case, if we had been asked the specific cell division, we could have said mitosis, because through mitosis we form two daughter cells identical to the parent. So the answer here, whereby cell M becomes cell N, that is cell division. Name the process, Roman 2. Name the process whereby cell N develops to form cell P or Q. That is cell differentiation, cell modification, or cell specialization. That is where this cell is going to be modified to form another cell of different kind. Part B of the question is asking, say the changes that could have occurred in cell N for it to be transformed to cell P. So changes could occur to cell N for it to be transformed to form cell P. So what changes occurred in the body of this cell? So you see that there was the formation of a new sap vacuole must be formed for this process to take place. Another reason we can say that the cell must enlarge, because we can see that cell N is small, cell P is large. So cell enlargement is another thing. Apart from that, formation of chloroplast in order to manufacture food and nutrients to provide the cell with energy for specializing or modifying itself. Apart from that, you can see that the nucleus of the plant cell is found on the periphery or on the side. So by this you can say that the nucleus must also be displaced from the center to the side of the cell, among others. So the next question is asking, state 3 characteristics of apical meristematic cells. So what are the characteristics of the apical meristematic cells? So the first characteristic is that this cell must have a thin cell wall. So apart from that, they must have a mature nucleus in order to facilitate the process of cell division, in order to control the functions of the cell. And then for these meristematic cells, they are unique because they are plant cells which do not have a sap vacuole. So those 3, among others, are some of the characteristics of the meristematic cells. So one unique one is that they are plant cells, yes, but they do not have a sap vacuole. So the next question is asking, a follow-up question, letter B, is asking, in the root of seedling, which tissue develops first? Is it the phloem or the xylem? So in the roots of a seedling, which tissues develop first? The xylem or the phloem? So the phloem develops first in order to translocate food from the seedling to the growing root tips. That is it. So the phloem must develop first in order to translocate this food from the cotyledon region to the root tip, so that the root can be able to grow and anchor the seedling inside the soil before now the plumule begins to grow. That is now the xylem. That is now the xylem. Before now the xylem begins to grow in order now to transport water and mineral salts. So it's the phloem vascular bundle which must grow first. So question next is asking, say the significance of metamorphosis in animals. We have gone through this. What is the importance of metamorphosis? So metamorphosis reduces competition between egg, larva, pupa, and adult. So it greatly reduces this competition between these life stages. So what can these organisms compete for? So these organisms compete for mates, they compete for food, they compete for space, and also predation, like whereby you see that predators of that organisms are unable to feed on these organisms or that organisms. So generally competition is able to be prevented through the different stages of metamorphosis. So the next question is asking, the graph below represents change in dry weight of endosperm. As you can see, we have 2 graphs. So we have one going downwards, we have one emerging from down, going upwards. So I've been told that the graph below represents change in dry weight of endosperms and embryo of cereal during the process of germination. So question letter A, we are being asked, account for the change in dry weight of the endosperm. Account for the change in dry weight. How do you answer accounting questions? If you have been asked account, they want you to give a reason why something happens the way it has happened. That is accounting. Give detailed information why it has happened the way it has happened. So for this, account for the change in dry weight of the endosperm. So for this, you can say that the dry weight decreases. Why does the dry weight decreases according to the graph? So the dry weight decreases because the stored food is hydrolyzed and transported to the embryo and used to make energy for metabolic processes. That is it. So we have gone through this time and time again. That water is used to hydrolyze the food in the embryo. So as this, not in the embryo, in the cotyledon, as this food is hydrolyzed, so this food is slowly depleting because the embryo is using this food. That is why we have said that the dry weight is going to decrease also as per the graph. So part B of the question is asking, account for the changes in dry weight of the embryo. So account for the changes in dry weight now of the embryo. Of course, the dry weight of the endosperm will decrease because the food is being used by the embryo. But what about now the embryo? So the dry weight of the embryo is going to increase. Mbona? Because the embryo is the one which is using this food. So the dry weight of the embryo is going to increase. This is because the embryo develops new tissues and cells which lead to its growth. And that is it. So the dry weight of the endosperm will decrease, while the dry weight of the embryo will increase because the embryo is developing new cells and tissues as per its own metabolic processes which facilitate growth and development. So the next question is asking, name the structure that produces juvenile hormone and molting hormone in insects. So we have also gone through this. So name the structures that produce juvenile hormone and molting hormone in insect. So these structures which produce juvenile and molting hormone in insect, they are the prothoracic glands and the corpus allatum, which we have already discussed. So the prothoracic glands and the corpus allatum are the ones which are responsible for producing juvenile hormone. The first function of juvenile hormone, we said, is what? So juvenile hormone is the one which now inhibits the process of molting from taking place, among others. So the regions, remember, we have the prothoracic glands and the corpus allatum region. So part B of the question is asking, what is the importance of molting in insect growth and development? We have said this time and time again. So shedding of the exoskeleton leads to rapid cell division of the organism, whereby this cell division of the organism leads to growth of the organism. So through this, the organism is able to grow. So the next question is a diagram, we are being told. The diagram below represents a maize fruit. Yes, it's a maize fruit. Why did we say it's a maize fruit according to reproduction topic that we discussed? It's a maize fruit because on the lower side of the fruit you can see the point where it was attached to the placenta. On the top side you can see there is also a scar. The scar on the top side represents the remains of style. So whereby that is where the style had emerged from. On the lower side it represents the point it was attached to the placenta. So if an organism has got two scars, that is a plant organism. If it has two scars where it was attached to the placenta and the remains of the style, that is not a seed, that is a fruit. And the maize is called a maize fruit and not a maize seed. So this question is asking, the diagram below represents a maize fruit. Name P, R, S, and T. Those regions P, R, S, and T. So as you can see, the letter P is the outermost region, which is the testa. The letter R is the cotyledon, the letter S is the plumule, as letter T is the radicle. So the radicle which develops to form the first roots, while the plumule which develops to form now, which now emerges now to hold the young leaves to facilitate the process of photosynthesis for this plant. So that is that diagram of the maize fruit. Part B of the question is asking, which part will stain iodine blue black? So which part of this maize will stain iodine blue black? So for this part we have part Q and part R. So part Q being the endosperm and the cotyledon, yes. So the endosperm and the cotyledon, part R, are the ones which are going to stain iodine blue black. But why will they stain iodine blue black? Because they contain starch and nutrients for the developing embryo. And that is the region. So the answer to this is part Q and R. Even if you know that part Q is the endosperm and you know part R is the cotyledon, remember the question is asking you which part in the diagram that has been drawn, which part. So the part is part Q and part R. Even if you know Q is endosperm, don't say endosperm. Even if you know R is cotyledon, don't say cotyledon. Use what is given in the diagram. So it is part Q and part R. So the next question, part C, is asking, during germination, what happens to the structure labeled S and T? So the structure labeled S is the plumule and part T is the radicle. So part S grows upward to form the shoot, part T grows downward to form the roots, and that is it. So question next is asking, the diagram below is a section of a young plant. As you can see, we have those letter E, F, G, H, and J. So part of the question in this diagram is asking, State the class of the section. So what class does this section belong? This section belongs to class Monocotyledonae. Why do we say it belongs to class Monocotyledonae? Because you can see the vascular bundles are scattered inside the cortex. You can see, if the vascular bundles are scattered inside the cortex, automatically that becomes class Monocotyledonae. So part B of the question is asking, name part E and G. So what's the name given to the part labeled E? What's the name given to the part labeled G as per the diagram? So the part labeled E is they are the epidermal hairs because they are found in the epidermis. So you can call them, or you can name them epidermal hairs, and that will be right. So part labeled G part labeled G is the cortex, because cortex is the one which forms the ground tissues which involve the sclerenchyma and the collenchyma. So that part labeled G, that now forms the cortex part of this diagram. So question letter C. State the function of the part labeled E. Yeah, state the function of the part labeled E. Those are the epidermal hairs. So the part labeled E, just like the hairs on our skin, we can also talk about the same thing. So this part prevents moisture from leaving the plant section, thereby preventing transpiration from taking place. Thereby preventing excess transpiration from taking place. Because warm air is going to be trapped between those hairs, thereby preventing cold air from taking all the water that are embedded, or the water that are found on the surface of the epidermis. Thereby this part prevents moisture from leaving the plant section, thereby preventing the process of transpiration from taking place. Question letter D. Explain why plant may grow big or become woody. So explain why this plant, may grow big or become woody. So why do you think this plant may grow big, very large tree, or become woody? So this is because there is no vascular cambium which leads to secondary growth. Or the question is asking, explain why this plant may not grow big or become woody. So explain why this plant part may not grow big or become woody. So this is because you can see in this diagram, there is no vascular cambium.
Biology.